Hi students, last class we discussed about transportation of gases. How the gas is transported by the blood, oxygen mixed with the hemoglobin to combine oxyhemoglobin. And while they are climbing the mountains, when they are going into the seas, they will carry the oxygen cylinders while reason also is up. And we discussed about mitochondria, the currency of the cell, AT, adenosine triphosphate. Also we discussed about glycolysis, aerobic respiration and anaerobic respiration. Today we are going to see some experiment with the yeast. In 8th class, in 9th class, we did so many experiments with the yeast. So this is second lab activity. The first lab activity is oxygen produced during photosynthesis in the presence of light. So this is the second lab activity. It's a very important for 4 marks. I think only 4 or 5 lab activities is there in the entire magazine. So this lab activity, 99% we can expect one lab activity. So whether it is 1 or 2 or 4 or 5, whatever may be. So today we are going to see that some experiment with the yeast. How anaerobic respiration takes place when the yeast is mixed with the glucose. Which type of gas is going to release? What happens when the gas is released in this experiment? We are going to see that. So we can see the simple diagram. So in the lab activity, this diagram is the most important. We have to compulsorily write this diagram. Experiment with the yeast. Come, take one jar. And you have to take one jar. It should be made up of the glass. You have to insert one thermometer. What happens? Why we should use this thermometer? We can see the temperatures in the thermometers. We have to take another tube also. How should we know whether the gas is going to release in this experiment? You have to connect this another tube. With another experiment, it's containing with lime water bicarbonates. So how many materials, what materials is required? One conical flask, bell jar, which one made up of with the glass, conical flask which we use, bell jar. Thermometer should be taken. Thermometer. Yeast in boiled and cooled glucose. This is paraffin. Thermometer. Derivative. This is lime water. Lime water. Come to the explanation. Aim is experiment with the yeast. How the carbon dioxide is going to release in this experiment? The material is required: conical flask, thermometer, delivery tube, another beaker with the lime water. We need a liquid paraffin. We need yeast and glucose. Yeast and glucose. Come to this yeast. This yeast we can prepare. We can use this yeast in the preparation of alcohol, in the preparation of wine, in the preparation of beer. In bakery also they will use this yeast. In the preparation of dosa, in preparation of idli, bonda, we will use this yeast. These are useful for us. These are useful microorganisms. Yeast is useful microorganisms. Come to the explanation. Come to the explanation procedure process. Set up this like an experiment. Take the glucose first. Take a glucose. We have to boil it. The glucose. Then cool it without shaking. Take the glucose. We have to boil the glucose. Why we are boiling the glucose? To remove the oxygen. To remove the oxygen present in the glucose. To remove the oxygen present in the glucose. Why we are removing? Because this is anaerobic respiration. Anaerobic respiration. Anaerobic respiration is nothing but absence of oxygen. Fermentation is called as anaerobic respiration, a low amount of oxygen. To remove the oxygen present in the glucose, we are boiling the glucose, then cool it without shaking. After cooling the glucose, we have to add yeast into the glucose. We have to add yeast into the glucose. So we have taken glucose, we are boiling to remove the oxygen present in the glucose. Then we are cooling without shaking. Then we have added yeast into the glucose. So it is okay. We have removed the glucose. We have removed the oxygen from the glucose. If any oxygen entered into the glucose during this experiment, that's why do not enter any gases into this glucose. We are adding a liquid paraffin. Why we are adding liquid paraffin? Because there is a chance to enter oxygen into the glucose. If we have added liquid paraffin on the layer of this glucose, if one layer of this glucose liquid paraffin, the oxygen, it will prevent the entry of oxygen into the glucose. We are adding liquid paraffin. How should we know after boiling the glucose if there is an oxygen is present? Our experiment will be failed. That's why we have to do one more test. If whether oxygen is present or not in the glucose, we have to add few drops of niacin or Janus green bee. On the solution, on the given solution, before adding yeast, take a small portion of glucose. Then we have to add Janus green bee. What happens? From blue color, from blue dye to it will turn into pink color. When we have to add the Janus green bee into glucose, so the blue dye of glucose will turn into pink color if it is turned into pink color, then we consider there is a low amount of oxygen is present, or oxygen is not there. High amount of oxygen is not there. If it is not turned into pink color, then we have to boil it to remove the oxygen. So this is the test to identify whether the oxygen is there or not in the glucose. Oxygen is there or not in the glucose. Now come to the experiment. So boil the glucose, then it is cooled without shaking, add yeast, add one layer of liquid paraffin to prevent entry of oxygen into the glucose. We have inserted the thermometer into the glucose. Then we have collected another delivery tube with another test tube with the contained with the lime water. Then start observing this experiment. Observation of this experiment. What happened? After few hours, the reaction takes place between the yeast and the glucose. The reaction takes place between the yeast and the glucose. How can we know? So, in the thermometer, we can see the temperature is going to increase. The temperature is going to increase. So, the energy is releasing when the reaction takes place between the yeast and the glucose. The energy is released in the form of heat. When cellular respiration takes place utilizing oxygen, what happens? Carbon dioxide, water and energy is released. In anaerobic respiration, what happens? Lactic acid is released or ethanol is released. Ethanol, carbon dioxide and energy is released in the yeast experiment in the absence of oxygen, that is anaerobic respiration. How should we know carbon dioxide is released or not? That's why this delivery tube is connected to the lime water. When the carbon dioxide is released in the experiment, this lime water will be converted into white precipitation. So, during this process, carbon dioxide is released. How should we know? When the lime water is converting into milky white precipitation, then we consider carbon dioxide is released. How should we know energy is released? When the readings points are increasing in the thermometer, then we consider energy is released in the form of heat. So this experiment will show that when the reaction takes place between the yeast and the glucose, energy is released in the form of heat. Carbon dioxide is released. When lime water converts into milky white precipitation, then we consider the carbon dioxide is released in experiment. Very simple experiment and very important for 4 marks question. So why we are taking the yeast? So we are taking glucose. So glucose, boiled glucose. Then we are cool. Why we are boiling? To remove the oxygen present in this glucose. Then we are cooling. Without shaking, we are adding yeast, then we are adding liquid paraffin to prevent entry of oxygen into the glucose. How should we know whether the oxygen is present or not in the glucose? We are testing the diazine or Janus green B. Add few drops of Janus green C in the liquid. So it will convert blue dye into pink. If it is converted pink color, then we consider low amount of oxygen is there. It is not turned into pink color, then we consider high concentration of oxygen still there, we have to still again heat it. Then finally we release carbon dioxide. This is a very important and easy experiment. Some experiment with the yeast. So one more experiment is there, that is the activity. Observing changes during combustion of glucose. Observing changes during combustion of glucose. What changes we can see when the glucose is burned? What changes we can see when glucose is burned? In a cellular respiration also glucose is burn, breakdown, carbon dioxide, water and energy is released. Here also what happens when indirectly in the lab, when we burn this glucose, what happens you can see. Again, here also connected to the delivery tube, another test tube that is called as lime water. I'll take one burner here. Test tube. Delivery tube. Lime water. Stand. It's a burner. In the test tube, we have taken glucose. So, observing changes during combustion of glucose. When glucose is burned, what happens? Which type of gas is released? So, take this experiment like stand, burner, test tube, delivery tube, another test tube, another beaker, continue with the lime waters. In our cell also, in our body also, when cellular respiration takes place, glucose is going to break down. Glucose is going to burn. What happens when the glucose is burned? Our body is not entirely damaging or destroying or burning. What could be the reason? Look at this experiment. So we have taken glucose into the test tube. Continue with one stand. We are starting burning this glucose. So it's connected to the other test tubes, continue with the lime water. So this glucose as a flow, we can take a sugar also. We can burn the sugar. Sugar are in the form of crystals. When we are burning, what happens? When we are burning this glucose or when we are burning this sugars, what happens? This crystal started converting into the crystals will convert into the liquid forms. They are converted into liquid forms. When they are converting, some energy is going to be released in the form of heat. So we can see some air bubbles on this test tube. When it's converting, when it's burning, the energy is going to release. So this energy is in the form of heat. When heat is releasing, so we can see some smokes on the test tube. Some small air bubbles are evaporation while the burning of the glucose or sugars. Those are nothing but heat is nothing but energy is released. So when glucose is burned, it's releasing, it's utilize oxygen and release some energy. So when this energy, when it's release some gases, it's release energy and it release gases. When these gases are mixed with the lime water, when the glucose break down or burn, it releases energy. It also releases some gases. When it gases is released by the breakdown of the glucose, when it's combined with the lime water, when lime water converted to milky white precipitation, then we consider carbon dioxide is released. When carbon dioxide mixed with the lime water, then it will be converted to white precipitation. Milky white precipitation. So this observing changes during combustion of glucose. What can we observe? When glucose is burning. So what happens? Converting into liquid. Then energy carbon dioxide is released continuously. Once the glucose started burning, we cannot stop it. It will be converted into ash. Finally, it will convert into ash. So there is no energy. Energy is released. Gas is released. How can we know? You can see the bubbles. How can we know the carbon dioxide is released? When the lime water converted into milky white precipitation, then we know that carbon dioxide is released. But once the glucose is burned, we can't stop burning of the glucose. But in our body also, the glucose is burning. But why the body is not damaged? Why not burning? Why cells are not burning? To control the process of burning in our body or cell, we have sufficient water in our body. So this water is going to control the burning of the glucose. It is required, then it will break down, then it will start the burning of the glucose in our body. So these are the important. So both are important, but the mostly important is so some experiment with yeast. Anytime, at least they'll ask 1 mark question finally from the experiment of the yeast. So this yeast we can see in the fruits also. You can see fruits, vegetables. This yeast we can see. So this yeast experiment is very important for 4 marks. Or else they will give the diagram and they will ask the labels. So observations, precautions. So the very important for 4 marks. Today that's why I am saying only 2 experiments. One is the yeast experiment observing of changes during combustion of glucose. Just it is very easy. Both are easy. Just you once read and write without seeing. Yeast with experiment, diagram and explanation. Read once the workbook is there in your workbook. So you have to read this in both experiment and write without seeing, students. Okay, thank you.